Thank you. So as Terry said, or mentioned, I'm Patrick Hanley from Children's National, and my role is actually to be the laboratory facility director. So the place in which we make all of these wonderful therapies is my job. I run that. So we don't make the cells for Terry, unfortunately, but we have our own manufacturing facility at Children's National, which is actually built with the help of Shirley Howard and CCF. And I'm also the Next Gen Award winner, so thanks to CCF for supporting me in all of our work. So before I begin, I give a lot of talks about how to run a cell therapy facility, and what I realize each time I talk is that there's more and more and more people to thank, and more people who are actually involved in making these therapies happen. So it's not just the wonderful team I have here who is always evolving, but in fact, there are other people who are involved that work behind the scenes and perhaps don't get as much mentioned. So for instance, we have a good laboratory practices facility that does a lot of our release testing to make sure that they are the cells we want, that make sure that they're not contaminated. We work with lab medicine to help send off the infectious disease marker testing to make sure that we are using eligible donors so we're not transmitting viruses to the patients we're trying to treat. The blood bank helps us with some of that same testing. The flow lab, again, makes sure that we have the right cells. Our clinical research team helps to enroll the patients. They consent the patients, and then they help us plan the follow-up testing. We do the infusions of our patients. About half of the patients that we infuse at our hospital actually come from other hospitals like Johns Hopkins or from CHOP or the NIH. And we're able to do that because we have a CTSI where we can infuse these patients. The HEMONC BMT unit refers patients to us. And one perhaps mostly unrecognized unit is actually the environmental services people. So if you can see back on our last slide, we run a clean room. Well, this clean room needs to be cleaned, and it's cleaned by that environmental services people. And they do a great job. We have to train them ourselves. And nobody says, hey, you did a great job emptying my garbage today. But I really want to acknowledge the great job that they've done cleaning our facility, because when I started, it was problematic, and they've really worked hard to make sure that that clean room is kept clean. And also, you'll notice here that our picture was taken in front of the CCF logo because, again, you guys did help us build that facility. And so what do we currently offer at Children's National? Well, I think our bread and butter is really working to manufacture CMV, EBV, and adenovirus-specific T cells. And the reason we want to make these are viruses, by the way, and the reason we want to make T cells that target viruses is because after a bone marrow transplant, one of the biggest causes of death are viral infections. And so if we can target those viral infections, then we could perhaps increase the survival of about a third of those patients. Another option is LMP1 and LMP2 specific T cells. So these are just proteins that are expressed by the virus EBV. But what's remarkable is that EBV is associated with lymphoma. And so if we can target the virus EBV, we can actually treat lymphoma using these T cells. Another option is similar to what Terry was just talking about, where you can make these leukemia-specific T cells, but instead of genetically modifying them, we can actually just grow them up in the lab by exposing them to proteins that are expressed on the tumor. And so that's what this trial is, just expanding T cells that target tumors. Another option instead of a bone marrow transplant is a cord blood transplant, and we can actually manufacture virus-specific T cells from cord blood, which is interesting to your point because these are naive T cell derived. They're not derived from the memory population. And so there is perhaps some difference in efficacy or persistence because they're from the naive population, not the memory population. And that's something that we're currently investigating. What's unique is this is personalized. This is the epitome of personalized medicine. We make T cells for you. But another option is we can make T cells for Jeff. And then, if he has a similar HLA type, or just like blood typing, we all have an HLA type. If the other Jeff needs those T cells, then perhaps we can give those T cells to him as well. And that's the reason this is advantageous is because they're available immediately. So you have an infection, we can ship those cells to Hopkins that night and treat the patient. And so that's really an advancement in this T cell personalized therapy. We just started manufacturing autologous HIV-specific T cells. Because we think that we can target the HIV and deplete the reservoirs. And so that's currently ongoing at UNC. And lastly, we can manufacture mesenchymal stromal cells, which are a type of a type of cell that instead of attacking things says, hey, hey, whoa, whoa, don't attack me, just kind of relax. So it can be used in sort of autoimmune diseases. So I just wanted to give you a brief flavor of what the therapies that we have ongoing in our facility here, and then I'm going to actually expand upon one or two of these. So I mentioned a bone marrow transplant. What is bone marrow transplant? Well, it's a treatment for relapse and high-risk malignancies, as well as many genetic disorders. In fact, it's one of the only curative options. Unfortunately, if you're getting a bone marrow transplant, what that means is you're going to deplete. So say I'm the patient who needs a bone marrow transplant. You need to somehow deplete my bone marrow, the stem cells in my body, and replace them with someone else's. And to do that, you need to give them agents like chemotherapy, total body irradiation, and T-cell depletion. And the result of that is, well, you don't have an immune system for some time. So after a transplant, the three biggest causes of death are viral infection, shown here in black, which I already mentioned, tumor relapse, which Terry has talked about, as well as other things like graft versus host disease. And what that is that's when the graft that you put into the patient actually attacks the body. And that's where those mesenchymal stromal cells will come in handy, because you can tell the body not to attack the cells, or tell the cells not to attack the body. So what we try and do using cellular therapy is attack each one of these problems, and by solving them, hope to provide a treatment for all of the patients receiving a bone marrow transplant and ensure 100% success. So why do we need other options beyond antiviral pharmacotherapy? Well, antivirals work most of the time, so that's great. But unfortunately, there's only a few of them. And if they don't work, we don't have any other options. And this has never been more clear to me than since I started my role at Children's National, because we now receive probably 50 to 100 referrals a year for patients who have failed these antiviral therapies and do not have any other options. And therefore, they call us saying, hey, can you please make us these cells, or do you have any other options for our patient who has this viral infection? So what we do is we ex vivo expand antiviral T cells. We have these cells in our body already, and we just expand them to target the viruses and then give them to the recipient. And this is a schematic of what the bone marrow transplant is now. So you harvest the bone marrow, we process it in that clean room facility, and give it to the patient who's been already myeloablated. What we want to do is we then collect blood, expand the immune cells to target the viruses, like I told you, or to target the tumors, and then give them back to the patient. This is a protocol that's run by Dr. Keller, which is what I just told you about. You know, these donors already have the viruses in their body, the T cells for the viruses in their body, so it's easy to expand them. We have a phase 1 trial open at Children's. We can treat as prophylaxis or as treatment, and we treated 16 patients. One patient was treated with donor T cells that I mentioned before. 10 of them were treated for CMV, 4 for EBV, and 2 for adeno. Those are just different viruses. And this is what it typically looks like. You see in red here, this is the viral count in the patient. You see here that we gave a T cell infusion here and a T cell infusion here. And shortly after the second T cell infusion, there was a decline in the viral load. And then you can see the detection of T cells recognized as a virus in the patient. This is more clear here, I think. This patient had an EBV infection. They received the treatment for EBV, the rituximab. And you see here in red, this is the viral load of the patient. The viral load's not going down despite the therapy. We gave them EBV-specific T cells, which is shown here in blue. And you see there's a coinciding decline in viral load and elevation of the EBV-specific T cells inside the body of that patient. And this is what we typically see when there's clearance of the virus and the presence of our cells. So overall, we've treated 16 patients. We've had 9 complete responses, so the virus is completely eliminated. 3 partial responses, a 50% decline in the virus. And 1 patient, unfortunately, did pass away and did not respond. Interestingly, that patient did not respond to a second T cell therapy either. So overall, there's about a 92% overall response rate. So what do we do if the donor is not available? So this is great if it's a match-related donor, so the brother is the transplant donor. But what if we don't know who the donor is, or if they're not available? Well, that's what I told you about before, where we're hoping to create a bank of these cells. So this is another trial that's open at Children's, and we've treated 7 patients. And this is an example of one of the patients who was treated. Again, you see in red, that's the viral load. The patient received T cells. There's a very rapid decline in viral load. And again, we can detect the virus-specific T cells in the patient after the clearance of the virus. So overall, we treated 7 patients. There were 2 complete responses, 3 partial responses, and 2 that did not have a response. So that's about a 71% overall response rate. I'd like to note that 3 of these were treated as an EIND, which means that we're not the holder of the clinical trial, but we helped support another hospital make their own clinical trial so that their patient could be treated. So what's the summary here? Well, my point is just that it's feasible, it's safe in a phase 1 trial, and it does seem to be potentially effective. Now, the caveat is that, again, this is a phase 1 trial, so we're not looking at efficacy. It's not powered to assess efficacy, but it does seem to help our patients. All of these products have been manufactured at Children's for each patient. So that, again, that is the epitome of personalized medicine. And in total, we've treated 20 patients with these virus-specific T cells. Over the last 2.5, 3 years, we've treated about 50 patients with our T cell therapies. So what if the donor is not seropositive? What if they have never had the virus before, so they don't have circulating cells in the body that recognize the virus? Well, another option is the ex vivo expansion of the naive T cells that I talked about before. So the naive T cells are present in cord blood, umbilical cord blood. I mentioned that an option beyond bone marrow transplant is cord blood transplant. But the problem with cord blood transplant is that the rate of these viral infections are actually higher because they don't have circulating memory T cells to protect against the viruses. So can we expand virus-specific T cells from cord blood for use after cord blood transplant? I'm going to skip all this because it's basically just how we manufacture the cells, but basically we just take proteins from the viruses of interest and use them to stimulate the T cells. And we could not do it the same way as before because these T cells were naive. They're antigen inexperienced, so we had to really give them an extra boost in the priming. This is what we call an interferon gamma ELISpot assay. What it is it just measures whether our T cells are specific, whether they get activated when they see the virus. And we see here that these T cells do recognize 3 of the viruses. Skip this part too. What's interesting is when we made these cord blood T cells, so they're from cord blood, right, which is collected basically at birth. So no one had been able to make cord blood virus-specific T cells before. And so one of the critiques was, well, how do you know you're not just expanding cells from the mother and you're actually expanding the cord blood cells? So what we did was we did what's called fluorescence in situ hybridization, which basically means that we can tell whether a cell is derived from the mom or the baby if the baby's a boy. So here we had a male neonate, the male cord blood, and so we expanded the cells and then we did the staining for the sex chromosomes, and you can see here that there is a presence of a Y chromosome, which means that they're from the boy and not the mother. I won't go into this too much, but basically these cells are different than cells we make from the memory T cells. So if you were to make cells from me and then make cells from a cord blood, they're different in terms of what parts of the proteins they recognize. And so there seems to be a differential immune response to the viruses in the primary response versus the secondary response. And this has been reported in other viruses like HIV and EBV, but nobody's been able to make T cells for CMV from a cord blood before, so this was novel. So the summary of the cord blood T cells is we can do this. They're derived from the naive T cells, not the memory T cells, which again was unique. They are functional and they recognize atypical epitopes of CMV. But the big question is, well, can we use this in a clinic? Will they work? So it's great that we can make them in the lab, but what about in the clinic? So we did open a trial. This was first done at Baylor College of Medicine, and then we subsequently treated 4 patients at Children's National. 13 patients have now been treated with these T cells. 2 of them are awaiting infusion. I don't want you to look into this too much, but basically my point here is just that most of these patients were on some sort of GVHD prophylaxis at the time of T-cell infusion, which could impact the function of the T-cells. Now, one of the limitations of a cord blood transplant is that it's a cord blood that's delivered, so there's a finite volume of blood, and so there's only so many stem cells that are present in that blood. And so sometimes the engraftment of the new immune system is delayed. And so the way we manufacture our T cells is we actually take 20% of that cord blood unit. And so if we're taking 20% of it, are we going to further delay engraftment? Well, the answer seems to be no. When we did a study of time to engraftment in patients receiving the whole fraction or patients receiving 80%, actually the patients receiving only 80% engrafted faster. I don't think it's because it's a better transplant. I think it's just because in order to be enrolled in our study, we had to have bigger cord blood units with a higher cell dose. There's been no toxicity to date and no GVHD associated with the T cells. And by now you're familiar with this graph. Basically, the patient had EBV shown in red. After the T cell infusion, we see an elevation in the number of EBV-specific T cells that coincides with the viral decline. This is an adenoviral infection, and basically what I'm trying to show is only in one patient were we able to detect T cells that recognize adenovirus, and that's a patient that had adenovirus infection. So these T cells don't seem to be great at persisting if there's no antigen present, which I think is probably similar to what Terry showed. And then lastly, this is a patient that had CMV infection. You see here that despite the first infusion of T cells, the virus still continued to increase in viral load. But when we looked at the CMV-specific T cells in the body of that patient, what we found was that it was actually, the number was increasing already before the infusion of the second infusion of T cells. So in summary, 8 of these patients refrained free of infection, and of the 5 that were infused, all of them did seem to have a complete response eventually. And so it does seem to be feasible, although it's much more difficult to make these cells than the memory-derived T cells. So do they persist? One thing we can do to check persistence, since we can't follow these, they're not genetically marked like Terry's, but what we can do is we can look at the T cell receptor genetically and then use that T cell receptor as a barcode to follow it over time. And that's what we did here. You can see that a patient that had no reactivation, there were a couple T cell receptors that we can detect at later time points, but there are more T cell receptors in the patients that did have some sort of reactivation. And in the patient that had the CMV reactivation, you can see that upwards of 15% of all the T cell receptors in that patient's body were derived from the infused T cells. I'm not going to go into this because I think we're running low on time, but basically this has progressed into what my CCF next-gen project is, okay, well, we can make T cells from umbilical cord blood, which is a naive T cell source, but what about when an adult donor like myself is CMV seronegative, so antigen inexperienced? I don't have CMV, which is between 40 and 60% of the population. And so we're using that same technology to isolate the naive T cells and then expand the cells. And I won't go into that. But I will say that these cells do seem to recognize the different parts of the protein, just like the cord blood cells, indicating again that the primary infection does seem to be different than the secondary infection. So in conclusion, there is an inherent difference between the naive memory response in vivo, or in vitro, excuse me. The epitope recognition from the naive population does seem to be dependent upon the precursor frequency. I didn't show that. But basically, when we search for the T cell receptors from the naive population, they were much more common than the typical precursors. We've now extended this to CMV seronegative donors, and we have an open clinical trial to test those. So again, I'd like to thank everyone who's been involved in this. Dr. Ballard is the sponsor of these studies. Of course, CCF has provided funding to take this to the next level and extend that research. I'd like to thank our EMT chief, Dr. Jacobson, as well as those who work in the lab to help make these cells available for the patients. Thank you.
Comment, there was no GVHD. Why isn't there any GVHD? There's no particular selection. With this antigen selection, but it's not perfect. Right?
No, of course, but so first of all, I think that these cells, well, they've been expanded. So we test them for alloreactivity before we release them. We do. With that said, we've never seen it. So we just take recipient-derived activated T cells, we don't pulse them, and mix them with the T cell line, T cell product, and assess cytotoxicity, and we don't see it. I presume it's because there's such a rapid expansion of the T cells that recognize the viruses that there's no T cells left that would recognize the host. But yeah, and we also use very low cell doses. So we infuse 5 million cells per meter squared to 20 million cells per meter squared. So it's pretty low. But yeah, no, we don't really see GVHD. In the mismatch setting, in the third-party cells, occasionally we will see grade 1, but it's probably not due to the T cells. It's a very complicated situation after BMT. You don't know it's from the T cells or the transplant. Yeah. It's true. Mm-hmm. Yeah, so what we did was we took 5 HLA-A2 cord blood units and selected the CD3s, and then we just sequenced all of the T cell receptors from the cord bloods. And we queried for all known typical A2 TCRs for CMV and the atypical, which were the ones who described those. Interestingly, only in 1 out of the 5 cord blood units were we able to detect the typical epitope. It's called NLV. And it was at a very, very low frequency. Whereas the other atypical epitopes, we found them in all 5 cord blood units, and in multiple cord blood units, we found multiple TCRs. So I don't know off the top of my head what the frequency of those TCRs were. I mean, it's in the STM paper we published, but yeah, it was really remarkable to see that. So it seems like there's just some sort of selection that happens, whether it's the cell tropism, you know, they're first infected in an endothelial cell or something, and then they, you know, at reactivation, they're presented by monocytes. Maybe that's the difference. I'm not sure, but yeah, I don't know the frequencies off the top of my head though.
With patients that have very high viral load, do you see any like cytokine storm type syndrome, or in fact things like worsening of pneumonitis or hepatic problems?
Yeah, no, so we don't. Interesting, I mean, I don't know the exact reason why we don't see GVHD or we don't see the cytokine release syndrome, but we do not, whether it be in the virus-specific T cells or, you know, I mentioned that we have tumor-specific T cells. Even in that setting, we do not see the tumor lysis syndrome. If you had a big, bulky tumor, solid tumor perhaps, I don't know if we would see that, but I don't know if it's the affinity of the TCR versus the chimeric antigen receptor or what it is, but we don't see that at all. And other people who use those same endogenous TCRs, I don't think that they see cytokine release syndrome either. So, and also Baylor, they don't see cytokine release syndrome in their CARs. Granted, they don't see the same efficacy. So